Welcome back to our Vitrolife Group Academy studio. We have the last session of the day, and it is my pleasure to introduce you, my friend and colleagues, Shannon and Angela, that I'm going to talk about biopsy and tubing best practices. So, Angela, Shannon, whenever you want, please.
Hi, I'm Shannon Kirkpatrick. I'm Senior Embryologist in Scientific Support with Vitrolife.
And I'm Angela Linville. I'm a Scientific Advisor with iGenomics. And today we'll be talking about best practices with tubing. So we'll break up the talk into two different parts. I'll first talk about the genetic testing portion and different types of no results, and Shannon will then take it over with the loading. So why is the loading procedure so important? Poor loading can lead to failed amplification and no results, requires rethawing, biopsy, refreeze, and causes additional stress of the embryo. Poor washing can lead also to contamination, and contamination can lead to a potential misdiagnosis. So there's two different types of no results, which can have different root causes. The first one is a no DNA detected, and the second is a non-informative. So for no DNA detected, this means that there was no DNA available for amplification, so we were not able to get any information at all from the sample that was sent to the testing lab. For non-informative, this means there was DNA amplified, yet the results failed QC, and this is typically because the DNA is not high enough quality for us to provide a result. So the different causes of these types of results: for no DNA detected, it's lack of nucleated cells, no cells in the tube, cells not properly placed correctly, must be at the bottom of the tube. If they're on the side of the tube, they can be missed and not amplified. If they're adhered to the side, it is not available for amplification, which can lead to a no DNA result. And then it can also be stuck in a bubble and not in the buffer itself, which also can inhibit or prevent us from amplifying cells. For non-informative, it can be if there's a large volume of buffer. This dilutes the enzymes, resulting in an insufficient RNA replication. The cells damaged during the biopsy or cell lysis can cause a non-informative. Poor quality cells or poor quality embryo. This is usually from DNA fragmentation, and also minimal or degraded DNA. So in most cases, we may never know the exact reason, but these give ideas to troubleshoot in your lab if you have if you have either one of these results. So a really important thing to consider when you are working with your PGT laboratory is to review the protocol, because every testing lab has different requirements. So they have different buffers, they can have different tubes, and also loading requirements, either dry or wet. It's very important that you follow the protocol from your laboratory to get the best results. And Shannon will then take it away with the loading.
Thanks, Angela. So let's start with some tips and tricks. Going back to our dish setup. So for biopsy, we want to set up our dish with small drops, maybe 10 to 20 microliter drops, one for each embryo to biopsy. We do want to be sure to label them appropriately, well labeled with patient information, as well as our embryo ID number. There are thousands of different ways to set up your biopsy dish, so follow your laboratory protocol. Just make sure your drops are not close enough where they're going to merge together. From there, we can add a PVP drop if we want to. We also want to make sure that we have various techniques of biopsy available to use. Every embryo is different, so every biopsy is different, and we may need to use different types of procedures or techniques depending upon each embryo. This may include our traditional kind of pulling, tearing technique, the old flick, which is the rubbing technique of the biopsy pipette on the hatching pipette. It may be the new flick or the tap, which has become everyone's favorite these days. And then seldom used, there is a technique using the oil droplet. So it's using the tension from the media drop and the oil to sever your cells from your embryo. What constitutes a good sample? We want to look for between 4 to 8 cells in our sample, and we do want to make sure that we see intact nuclei. So Zoom in onto your sample, see that there are 2, maybe 3 intact nuclei that you can see before taking your sample, and that's what we're looking for. You do want to make sure to complete all of your documentation appropriately, both for your laboratory and for your testing laboratory. That makes things easier on everyone. Do set some KPIs. So this might be for each individual technician. Keep track of your no result rates, your non-informative rates, and this will help see if anyone deviates from protocol and may need some additional training. With your hood, you do want to start off with a nice clean hood. It should be disinfected. You can use a UV light if your hood has a UV light, but make sure you take things out of your hood before doing that. Acquire all of your tools and have them laid out in an organized fashion and have them ready for your tubing. Of course, you want to wear sterile gloves, a mask, a hair cover. Label your tube appropriately. For iGenomics, this would be the patient's initials with the embryo number on both the top and the side of your PCR tube. You can either do preloading or dry loading. So what this means, you may take your biopsy sample and load it directly into your PCR tube, or you may preload the tube with maybe 2 to 3 microliters of your wash buffer all the way at the bottom. We do want to minimize the final volume of our sample, so do not preload with more than about 3 microliters, and best to stay as small as you can with that. But sometimes it does make it easier to have a preload sample or preload media within there. A microcentrifuge is a great thing to have on hand to drive that media down to the bottom of your PCR tube. Then we need to make sure to wash our cells. So 3 to 5 wash drops using our wash buffer will be set up into a dish. 1 row of wash drops per embryo biopsy sample. Do not go back into another row of wash drops with any particular sample and duplicate that. You do want to have an adequate size of wash buffer, so maybe 15 to 20 microliters per drop, not too small. And this will be performed at room temperature rather than at 37 degrees because we don't want to degrade any DNA now that it has been taken away from that embryo and it doesn't have that living structure that's supporting it anymore. Do go down to a smaller pipette tip when you're working with your biopsy sample versus what you would be using for your embryo. So a nice small pipette tip will help you keep control of that sample. And of course, change pipette tips between each sample. Don't reuse it for your next sample, even though it is the same patient. Washing is important because we want to remove any extraneous DNA, cellular debris, oil, any of those things. So we do want to gently wash the sample through every drop, maybe in and out several times through each drop, and then into the next drop, and so on until we get to our final drop and are ready to load. When loading, we can again do wet loading or dry loading. The important thing is we want to get that sample down to the bottom of that PCR tube. Of course, we can't really see into the bottom of the tube, so maybe just on the cusp, on that shoulder, is the easiest place to load where you can see your samples. But you do want to make sure that you visualize the cells entering the bottom of the tube. As soon as you visualize those cells, remove your pipette tip and don't expel the rest of the media. That will help keep that sample nice and small. Few things that we want to watch out for. Starting on the left, we don't want to load our sample on the wall of the PCR tube. As Angela mentioned, it can get stuck there and not get washed down into the bottom. We also do not want to have oil in with those cells. Oil can coat those cells and interfere with the PCR amplification process, and we may get a non-informative result. We also want to watch our volume. Remember to keep that total final volume under 5 microliters. Otherwise, our sample will get diluted, and again, we may have non-informative results. So nice small sample size at the bottom of the tube. That is what we're looking for, and a microcentrifuge comes in very handy at this time to make sure your sample is at the bottom. Wash, rinse, and repeat. So complete this process for all of your samples. When you are working with your tubes, do make sure they are well organized. So on the left-hand side is a good example versus on the right where we have just random placement of tubes. Have a spot where you have your unloaded PCR tubes and then move them to another spot, another row, once you have your samples loaded so that you remain organized. Freeze your samples as soon as possible once you're completed with your case. And you do want to make sure to leave your samples in the freezer for at least an hour before shipping them out. That way you can be really sure that they are frozen and stuck in place in your PCR tubes before sending them off for shipping. So in summary, always make sure to sample an adequate number of cells, again, approximately 4 to 8 cells. Rinse those cells sufficiently to make sure you're not carrying over any extraneous matter. Properly place it in the PCR tube at the bottom. Keep that volume to a minimum. And then finally, make sure it does spend adequate time in the freezer so that it doesn't get lost in shipping. Thank you so much.